everything was going fine until the fall of '08. And so things have been put on the back burner. But we're optimistic if things begin to stabilize and recover, we'll be able to hopefully in the near future announce that we're developing a corneal regeneration program here in Denver. So just one example of that I told you of how the field of stem cell research has changed so dramatically in the last 10 years. And in fact, this has only happened within the last 3 years. So I mentioned that we have the ability to reprogram adult cells back into a cell that looks very much like an embryonic stem cell. Basically what we do is we can just take a tiny biopsy from the skin. We put the skin cells in culture. And then we introduce what we call reprogramming factors. And over a series of weeks, we can reprogram those cells back into an embryonic state. To allow you to understand what this means, if you think about your desktop computer, it would be like taking your hard drive and stripping everything from its memory and starting all over again. So basically, we're stripping all of the memory and the genetic code of an adult cell that told it had to be a skin cell, and we're taking it all the way back to the same state that it existed when it was in the early fertilized embryo. So if you think about it, this not only gets around the ethical concerns of having to destroy an early fertilized embryo to obtain embryonic stem cells, it's actually even better because this is personalized medicine. We can take the biopsy from the patient that we want to treat. So just to show you how we envision using this technology, this is a little boy named Adam. He lives in Atlanta. I've known Adam now for several years. He arrived with his parents on March 29th. We took a biopsy of his skin. Adam was born with an inherited skin disease where his skin blisters very easily. And if you look at the palms of his hands, you'll see that when the skin blisters, he tends to develop a very thick, almost like a plaque. This is kind of a protective mechanism because the skin blisters so easily. So we're going to try, we know we actually were one of the groups that actually discovered the genetic basis of this disease. So we're actually going to try to correct this defect in Adam's skin cells. So on the next slide, I'll show you how we envision doing this. As I said, we start at the top. If we move to the left, or counterclockwise, we've already taken a biopsy. We've put his skin cells in culture. We've expanded them. And now we're in the process of reprogramming them with the reprogramming factors to produce what we call induced pluripotent stem cells, or iPS cells. Once we get these cells, we can grow these cells just like embryonic stem cells indefinitely. We can actually freeze them and store them. After storage, we can break them out and thaw them, and they're still viable. So since we can expand them indefinitely, since we know the exact cause of his disease, we basically can use a procedure where we flip out the bad gene and replace it with a good gene. Once we've done that, then we expand the corrected cells, and we've now developed the procedure where we can now differentiate those cells back into skin stem cells. So once we expand them, then we can return them back to his skin as an autograft. So now we don't have to worry about the rejection of a transplant because we start with his own cells. So this is a problem with using embryonic stem cells. We actually don't know if cells from an embryo of unknown origin is actually used to generate an adult tissue that then is returned to a patient. We suspect that actually the patient will mount an immune response, just as patients mount an immune response to organ transplants and would have to be on immunosuppressive therapy for the rest of their lives. So we believe that this technique will actually avoid that. So just to give you an example of how fast the field is moving, Kurt Fried is a clinical investigator out on the Anschutz campus. He spent his whole life working on Parkinson's disease. Kurt has now actually taken a biopsy from a patient that's 60 years old who has Parkinson's disease. He's actually made these induced pluripotent stem cells, and he's actually shown now that he can actually differentiate these back into dopamine-secreting neurons, and that is the defect that underlines Parkinson's. So I think you can see how applicable this technology is. We have others in our group that can actually differentiate these iPS cells into bone, muscle, cartilage, cardiac muscle, etc. So I think you can just let your mind imagine the possibilities with this new type of technology. So there are obstacles that we have to overcome. We have to be able to make these cells carefully, and we and others now, I think, have developed the procedures for doing that. We have to be able to differentiate the iPS cells back into the adult lineage that we want to use to correct a defect in a patient. As I've mentioned, we can easily, we've done this very efficiently for the skin, and Kurt has done this for to make dopamine-secreting neurons. So this is rapidly progressing as well. The other thing that really will be initially an obstacle is the cost, because in order to expand a patient's cells, we will have to have, as I mentioned for the cornea program, we have to have a GMP facility, a good manufacturing practices facility, where the cells are grown in a very tightly controlled, clean environment. We actually have 2 small facilities on the new campus that we can use for initially testing this approach, but ultimately we'll need to expand that into a larger facility. But I actually envision that there will be specialized centers of excellence around the country. Not every medical school will have a GMP facility and grow these cells. You can, a clinician will actually be able to take a biopsy from their patient, send it to a facility like ours. We can actually make the iPS cells, correct the defect, differentiate those back into the adult lineage, and then ship them back to the clinician to use with their patients. So I think this is where the future is going for this field. So now I'd like to turn to a relatively a new area that is really not regenerative medicine, but it affects potentially even more people. And that is the idea. We call this, when stem cells go bad, seeds that maintain tumors. It's the idea that tumors themselves may actually be maintained by a cancer stem cell population. So just to give you an example of, if we take the skin, I personally work on the skin not only from the point of view of inherited skin diseases, but also skin cancer. I'm sure many of you, like myself, have probably already had a skin cancer removed. Most of us tend to develop skin cancer as we age. We're constantly exposed to the sun. UV radiation can induce DNA damage, which would result in a cancer. If you look in the upper panel, the skin is an example of regenerating epithelium. You're constantly sloughing and turning over your skin. So you don't really realize it, but every 3 weeks you completely replace the outer layer of your skin. So if you think about that, the only cell that can actually give rise to a skin cancer is actually the adult stem cell that actually regenerates the skin over and over, because that's the only cell that resides there for life. So the concept that an adult skin stem cell is the target for DNA damage that ultimately will result in a tumor is a very well accepted idea. And if you think about it, this not only applies to the skin, it applies to the most common types of cancer that we get, epithelial cancers. So this applies to the oral cavity, esophagus, lung, breast, prostate, GI tract, bladder, etc. These are all epithelial tissues that have an adult stem cell that's constantly regenerate the lining epithelium in those organs. So this is a well-accepted concept. The next concept, that the tumor itself may actually be maintained by a cancer stem cell, is relatively new. And just to show you how we demonstrate this in the lab, here's the next slide. If we look at the panel on the left, this is the way that we actually isolate and separate and enrich for stem cells. So we can use this not only for normal tissue, but also for a tumor. And so we take a large tumor, as shown on the back of the mouse in the right panel, dissociate it into individual cells. We use a fancy machine called a cell sorter to sort out cells that have stem-like characters. And if you look in that little blue colored rectangle, you see that the percentage of cells that actually have stem-like properties in a tumor is actually very small, less than 1%. The rest of the cells don't. And now if you ask which cells can actually form tumors in a secondary tumor assay in a mouse,